Uh, Good morning, everyone. I hope you're all students present there. So we will start with the continuing session from the yesterday. Mhm. हाँ मेरी क्लास है स्टार्ट मैं पी लूँगा चाय हाँ जी So yesterday we were done with this part, I guess. So we done with all. We just have a repetition from yesterday what we did. I will do it in a fast way. So we were basically discussing the definition of an enzyme, the important terms that are used to for the activity of the enzymes. Then what are the active sites that we have worked, making enzyme substrate complex, then enzyme product complex, and product leaving the active sites. And what is the importance of substrate? Lockean model we have discussed. Apoenzyme, holoenzyme. What is the difference between them? Apoenzyme is in active state, but with the help of cofactor, it's become a holoenzyme which is active state. And these coenzymes are two types, coenzymes, and these cofactors are two types. Coenzymes is prosthetic group. Coenzymes are the with a loosely bond by the aposome by the non-covalent bond, whereas the prosthetic groups are the one with the non-protein component tightly bound to the apoenzyme by covalent bond. These enzymes are quite specific. Their activation, they mainly do the push for the first activation energy in order to make their action fast. So like this we can see without enzyme is this like this, and with enzyme it will be lowered. Then we have discussed the summary of these enzymes: complex or hollow enzymes, simple, uh, prosthetic groups, coenzymes. What are these things presence? Then mechanisms and action of enzyme. How what are the various mechanisms? We will discuss in our next slide about that. So enzyme, and we have discussed this enzyme plus S give rise to enzyme substrate complex. Enzyme substrate is giving to enzyme plus product, and then enzyme plus S. This is a summary of the whole enzyme catalyst reaction at the end. This is also the same thing discussed with the diagram here, showing basically you the transition state was also present between your ES to EP, and then your product is released afterwards. So that was, and before that we have discussed some basic history about enzyme engineering, why we should study, what were the basics, key facts that have been found out in that part. So yeah. So now let's go to the next part of our enzyme engineering, that is the nomenclature of enzyme. Good morning, Soham. Good morning. So let's start with the nomenclature of an enzyme. So here we will be acquainted with the nomenclature and classification. We will understand the enzyme and to know the basic concepts of allosteric regulations and understand the feedback mechanism. Mainly we will discuss all these parts. So this is our enzyme, and these are the active sites and substrates want to get attached to it. So here the content is written: nomenclature and classification of enzyme. So enzyme substrate complex, cofactors and coenzymes and proenzymes that we will be discussing in this chapter. So let's start with first how we can classify and give the names to these enzymes. So basically they are classified and we categorize into them 6 parts. That is your oxidoreductase, transferase, hydrolase, lyase, isomerase, and ligase. That we have discussed in our first slide. How much percentage they are used in industry, how much they are important, but we need to understand, these 6 parts. These like what are the importance from the reaction point of view in our body. So oxidoreductase, that is a reaction which is done in order to find out the redox reaction, that is your reductase and oxidases that are happening together, whereas the transferases, it's a reaction which transfer a group from one molecule to another. So in this, what we can see, first of all, in oxidoreductase, you have a lactate, and then you have a pyruvate. Yeah. Lactate has OHCH with COO negative and CH3 methyl group. And then there is NAD plus. Then there is a lactate dehydrogenase, which leads to formation of pyruvate. So it's a double bond here, and we just remove this hydroxyl group. So this is the reduction of your lactate is done. Then the transferases is the transfer of any one molecule group to another. So transminase, which mainly is known for the transfer of amino group, kinases for the phosphate group. So here PNMT will be transferring the methyl group donor to this. This is norepinephrine making to epinephrine. So methyl group being attached there. Then comes the hydrolases, which cleaves the bond by adding water. So they could be phosphatases, peptidases, lipases. So here we have three molecules of water. So we will cross them here with a triglyceride, and then we have glycerol and fatty acids at the end. Yeah, and then liases are the one which remove groups to form double bonds and reverse back double bonds. So they catalyze, remove the groups to form double bond. So here we can see there's a double bond happening in fumarate. Then in presence of water and fumarase, they make a malate. Yeah, there's a hydroxyl group and H group, and just catalyze and remove the groups to form double bonds. On the reverse, break double bonds. So decarboxylases synthesis. Then there is isomerases, which catalyze intramolecular rearrangements. So like epimerases, mutases. So basically, it's change the structure. So like a phosphate group is attached on this oxygen, so it will just move to the another oxygen, to the second carbon, to the first carbon instead of the second carbon. So in the presence of phosphoglycerate mutase. In the ligase name suggests it helps to join. Uh, it's catalyze the reactions to make or break bonds. So if there's a DNA strand there with the hydroxyl group and another side on the phosphate group, DNA strands or DNA ligase will help to join them together. So let's discuss them one more time. From the first, oxireductase, they help in the redox reactions of reductase and oxidases. So we have understood the example of lactate, which got this one, your H group is removed, 2H is removed within the presence of lactate dehydrogenase. Then transferases group known for the transfer of molecules. So norepinephrine in the help of PNMT, the methyl group has been transferred to the group to make it epinephrine. Then hydrolases, they cleave the bonds by adding the water. So like phosphatases, peptidases, and lipases. And lysases, which catalyze removal of groups to form double bonds. On the reverse, break double bonds. So like this is the double bond here, so it will break in the presence of fumarase and also presence of water. Then isomerases, they are known for making intramolecular rearrangements. So like epimerases, mutases. So in this case, phosphoglycerate mutase, the phosphate will move from this oxygen level to the second carbon atom here, making from 3-phosphoglycerate to 2-phosphoglycerate. Then ligases, which catalyze a reaction in which two carbon CS, CO, CN bonds are made or broken. So we have DNA strand with hydroxyl group, phosphate group with a DNA strand, and then you have your DNA being attached in this phosphate group. So both DNA different strands, they are being attached to each other. So now let's start with the next part. The nomenclature of enzyme. So in most cases, enzyme names are ending with "ase" at the end. The common name for hydrolase is derived from the substrate. Like if it is urea, we can remove A from the end and then we can replace it with ase. So like urease. Then there is lactose. We can remove O's, OSC from the end. Then we can replace with ase and it becomes lactase. So that's how basically we give the names to the enzymes. So other enzymes are named for the substrates and the reaction catalyzed. Then lactate dehydrogenase, pyruvate decarboxylase. So lactate dehydrogenase, um, that the name for the substrates and that reactions catalyzed. So if lactate is removing the hydrogen group, then it will be lactate dehydrogenase. If it is we are moving a carboxylate group, then pyruvate decarboxylase. We are moving a carboxylate group from pyruvate, then we became the enzyme name becomes pyruvate decarboxylase. And some names are historicals, which doesn't have any direct relationship to substrate or reaction type, like catalase, pepsin, chymotrypsin, and trypsin. So that doesn't have any meaning why their name are given like this. Now, effect of enzyme on the activation energy of reaction. So here we can see a reaction, like let's A with the numerical components A on top and B with small b. This is a reversible reaction. In order to this K equivalent, this is your equilibrium constant. The value will be, we will have the value of your substrate raised to power its numerical constant on top, and then A raised to power small a. So like your product raised to power B, reactant raised to power A, small a. So enzyme basically it helps to speed up the reactions by lowering the activation energy and changing the reaction pathway. So basically what is it doing? So the activation energy, the push up reaction that it needs initially to make reaction happen, it lowers that and it's make a new pathway for it, which leads to the production of product from substrate on a very low energy route. The energy route that is required in order to make product will be very less. And every chemical reaction that is characterized during this equilibrium constant, which is reflection of the difference in energy between small a and small b. So this is KQ. So we have seen the difference between your in these twos could be calculated by this formula. So what we were discussing in my first lecture, how this enzyme are lowering their activation energy. So this is the diagram explaining that. So this is the reactant A plus B, this is the product C plus D. Reactant A plus B, product C plus D. And this is the progress of reaction. So reactant A plus B, product C plus D. So the activation energy required without catalyst is quite high. Yeah, it's quite high. Whereas the in catalyzed reaction, it's quite less. It's quite less in the presence of enzymes. So that's the major difference between your uncatalyzed reaction and catalyzed reaction. So uncatalyzed reactions has a large activation energy, that is EA, which is seen on the left, and on the right hand side has lowered significantly because of the rate of the reaction as will load with the help of your enzyme. So what happen on the rate of reaction if substrate concentration is increasing in the absence and presence of your enzyme? If there's no enzyme is present, if you keep increasing substrate concentration, the rate of reaction will be keep going in linear way. But if you have enzyme present, then it will form a Michaelis-Menten equation, in which the rate of reaction and substrate concentration will make a hyperbolic structure, and it will reach at the maximum state with the Vmax, and this will be Vmax, half Vmax here, making a Km value at the bottom. So we can discover, yeah, it's a different thing that we will be corely studying soon. So rate of uncatalyzed reactions increases as the substrate concentration increases. So both are proportional to each other. Then enzyme catalyzed reaction, they show two stages. First, as we know, enzyme substrate complex, then slow conversion to product, and this reaction is limited by enzyme availability. So these three things are being taken care in this graph. That is, first enzyme substrate complex is made, then it's slowly converting to product, and then it's also limited by the enzyme availability. Now, enzyme substrate complex, as we have discussed this in my previous slide that we have discussed yesterday, we have just have a revision about this. That is, there are multiple steps while making your enzyme plus product. So enzyme plus substrate, in the first step, it make a enzyme substrate complex, right? Then there's a transition state after step 2. Then after the transition state, step 3, enzyme product complex is made. And in the step 4, enzyme plus product is made later on. So the reversible, all we can see is all these reactions, step 1, 2, 3, and 4. These are all reversible reactions that represent this enzyme catalyst reaction. So we have seen that first reaction is enzyme substrate complex, then making a transition state, and then making a enzyme product complex. So how this enzyme substrate complex is made up of is it's a part of enzyme that combining the substrate in the active site. So active sites characteristics includes, so as the substrate is attaching to the enzymes, so what are the characteristics of these active sites actually? Basically, it includes pockets or clefts on the surface of an enzyme, um, which have mainly these free alkyl groups, active sites. These are called catalytic groups. And the shape of your enzyme changes as soon as your substrate is being attaching to the enzyme. And it attracts and holds these enzymes for using weak covalent interactions. And then after the product is formed, then it will release them. And the conformation of the active site determines the specificity of enzyme. So like how enzyme is your, how much determined it is, how much specified it is, it's all being governed by enzyme substrate complex. Now lock and key model, and we will discuss about induced fit model. So here we have enzyme plus substrate and enzyme plus substrate complex. So enzyme plus substrate, they combine together, and then enzyme plus substrate, they make a complex here. So you can see there's some structural changes happen to the enzyme also. So that's why enzyme may fit exactly where it has to be. Enzyme makes some conformational changes in order to accommodate your substrate molecules. But when it doesn't happen, so it will fail, and it doesn't let it happen further. Then there's an induced fit enzyme model, in which enzyme active site is quite flexible, not like the locky model, and we can have a proper conformational changes in order to accommodate the substrate molecule. So in this case, sorry, I just said something wrong. Lock and key enzyme model, the conformational changes doesn't happen in order to fit your substrate, but in the induced fit model, your enzyme will do some conformational changes into its own structure in order to adjust your substrate molecule. Yeah. Now, how the transition state and product formation is happening. Basically, E plus S giving rise to ES. That's we have just discussed, just everything. But how does enzyme promotes faster chemical reactions? How does the enzyme is basically making these reactions faster? So as a substrate interacts with the enzyme, its shapes changes, and this new shape is less energetically stable. So this transition state that is being formed or enzyme product is quite unstable, not that stable. And it has this transition state features both substrate and product and falls apart to yield products which dissociate from the enzyme. So that's how this reaction is playing a major role in this. Why we have very few contents? If you have any questions, please do write in the chat box. I will be happy to reply you. So possible types of transition state changes that is possible in it. So first is this is an enzyme substrate is being made. This is enzyme and substrate, and this transition state enzyme substrate complex are made like this, and then the enzyme product complex they come together to release water. So this one molecule of water is removed and the substrate are joined together and it's make an enzyme product. Your new product is being produced. So like this, new reaction is there. So you have glucose and fructose, and then you have a sucrose, and the water molecule is removed, and there is a bonding between your two, making a new product as a sucrose. So that's also type of transition states that happen. So enzyme might bring two reactants into the close proximity and maintain proper orientation. So possible types of transition states changes. What are the possible ways that we can change these? This enzyme might modify the pH of microenvironment, donating or accepting a pH or H plus. So here we have cofactors and coenzymes. Here what we can see is you have a apoenzyme plus substrate, but as we don't have any cofactor, so it will not do any reactions. No complex will form. No enzyme substrate complex is formed. But once we have copper as a cofactor and attached to the functional enzyme with active binding site, so it will make reaction occur and enzyme substrate complex is formed. So active enzyme, it's a hollow enzyme, polypeptide portion of enzyme, which and the non-protein part is the cofactor prosthetic group which attached to this apoenzyme. So cofactors are bound to the enzyme for maintain the correct configurations like metal ions, organic compounds, and organometallic compounds could be added. Further, these coenzymes is required by some enzymes. So here we can see in the right hand side this reaction. That is, I hope your students are present, and please confirm me if you're all present. If you have no further, if anything I could help, please keep talking with me so that I could confirm that you're present and listening to me. Okay, I think it's working. So coenzyme, it is required by some enzymes, that is inorganic molecules bound to the enzyme by weak interactions by hydrogen bonds. So we can see that enzyme here with the coenzyme position to react with two substrates. So this is the coenzyme is being attached and there are two substrates, substrate 1 and substrate 2 with a functional group attached to it. So enzyme with coenzyme position to react with the two substrates. So coenzyme picks up the functional group from substrate 1. So, so this coenzyme and substrate 2 and substrate 1, they all come together, join, and the coenzyme transfer the functional group from substrate 1 to the substrate 2, and hence we have two different products. P1 doesn't have any functional group and your P2 has a functional group. So in this way, products are released from the enzyme. So in this reaction, the coenzyme is helping to move one functional group from one substrate to another substrate. So an organic molecules that bound to the enzyme by weak interactions or hydrogen bonds, and most coenzymes carry electrons or small groups, and many have modified vitamins in their structure. Some water soluble vitamins and their coenzymes. So we can see here that is thiamine, yeah, which makes decarboxylation reactions. Riboflavin, FM and FAD carriers of H atoms. Niacin, B3, NAD carriers of hydride ions. So like this, ascorbic acids, the coenzyme we don't know, but it helps in the proline and lysine and collagen. Biotin, biostin coenzyme in carbon dioxide fixation. So like so on. So let's have a 5 minutes break or 10 minutes break from here, and then we start with the allosteric enzyme. Yeah, let me just get refreshed. Just 5 minutes. We meet at 7:50, so that I could recover and start again. So now let's start with the new topic of allosteric enzyme. Is there any message? Not so important. Allosteric enzymes. So basically, they are the one which have many sites, and they could be act as a factor or could be reducer. Like they could be positive effector and negative factor. So both things are possible in the case of allosteric enzyme. So effector molecules that change the activity of an enzyme by binding at a second site. So some effectors which speed up enzyme reactions, they will be the positive allosterism, and there will be some which make it slow, that will be your negative allosterism. So here we can see that the enzyme in the first case here, it has a factor binding site and our substrates got attached, and if a negative feedback factor is being attached to it, and this is the will be attached to it, it will come and being attached on the back of it, so it will make the active site not working anymore, and the active site will close and no further action. So this will be a negative allosterism. Yeah, so the active site is there, but it will not let it attach, it will go attached to the part of a negative feedback inhibitor, because after the substrate was produced, so there were two products were produced. So one was a small, this green part, and one was a bigger one. The bigger one got attached to this site, it was fitting there and act as a negative effector, and it makes the further reactions to go, not making to inactive site and doesn't let it happen. So some example here, the third reaction of glycolysis, if you remember from your glycolysis, which places a second phosphate group on fructose 6 phosphate. So ATP is a negative factor and AMP is a positive factor of the enzyme phosphofructokinase. So you have this here fructose 6 phosphate, um, yeah, and this phosphate group is attached on top, and you have ATP on the right hand side. Then phosphofructokinase, in the presence of ATP, they make fructose 1,6 biphosphate. They make fructose 1,6 biphosphate and ADP molecule also. So in this way, the reaction as a, we can say ATP is a negative effector here, and AMP is a positive factor of the enzyme phosphofructokinase. If there has been AMP present, monophosphate, then might be a different reaction, but if ATP is there, then it's a different reaction. So ATP is making, doesn't let the happen the reaction further, but AMP will let it happen. And now after the allosteric inhibition, now let's talk about your feedback inhibition, that is your. So allosteric enzymes, that are the basics of feedback inhibition actually. So you can see this reaction, A giving rise to B, B giving rise to C, D, E, F. So product late at the end, we have seen this F product. So what will happen, F will serve as an inhibitor to enzyme 1, since all the products are being produced, but it will inhibit the activity of E1, so that means the feedback inhibition. So product F will act as a negative allosteric effector on one of the earlier enzymes in the pathway. So it's act as a negative, that we have discussed the first definition in this while starting about allosteric enzymes. So it will come and attach to the negative sites, the effector site of enzyme, and doesn't let the enzyme to be active and doesn't let the happen all these reactions go further. So this is your negative feedback inhibition, and there is a positive allosterism also, in which we see allosteric activation is seen in the binding of oxygen molecules to hemoglobin. So hemoglobin, it's actually composed of, if we see, 4 subunits binding to the oxygen molecules, making HbO8. So this each oxygen molecules, they make conformational changes to hemoglobin and increase the affinity for oxygen. So that the hemoglobin could transfer the maximum amount of oxygen from rich oxygen areas, that is to the lungs. Or also the release of oxygen molecules decreases as the human globin affinity for oxygens also decreases into the release in the tissues. So both things are possible. Now next, that is proenzyme of the digestive tract. Mm. So in a protein modification, a chemical group is commonly added or removed from the protein, and covalent modification either activates or turn off the enzyme. And the most common form of protein modification is addition or removal of phosphate group. And this group is located at the R group here. Sorry, from here we have to start. So proenzyme is basically, that is an enzyme uh, which are in the inactive form. So they could be changed to the active form by either proteolysis, that is your hydrolysis, removal of the water group from the enzyme, and we mostly need them at the active site in the cell. For example, pepsinogen is synthesized and transported to the stomach, where it is converted to pepsin in its active form. So these are various proenzymes. So some examples of proenzymes in our digestive tract: proelastase, trypsinogen, chymotrypsinogen, pepsinogen, um, procarboxypeptidase, and then most of their activators is trypsin, and the enzyme that are being produced are elastase, trypsin, chymotrypsin, pepsin, carboxypeptidase A. They all helps in our digestion of our body, digestion of our food. And there could be also some protein modifications that could happen in a chemical group of the other covalent bond could be added or it could be removed from the protein. So anything is possible. So the most common form of protein modification is addition or removal of phosphate groups. So this group is located at the R group with the free hydroxyl group. So like serine, threonine, tyrosine. So all these groups, they make things done like this. So serine, threonine, and tyrosine. There could be also inhibition of enzyme activity, which is possible because of the competition from another complex molecules. So there are chemicals that can bind to enzymes and eliminate or drastically reduce catalytic activity. So classify enzyme inhibitors on the basis of reversibility and competition. So it could be irreversible, reversible. So irreversible inhibitors, and reversible could be of two types: reversible competitive, reversible non-competitive. So we will discuss each kind of inhibition now one by one. So irreversible inhibitors that binds tightly to the enzyme and thereby prevent formations of the ES complex. So irreversible inhibitors that bind tightly to the enzyme and thereby prevent formation of the ES complex. So they just bind to the enzyme, these inhibitors, and doesn't let the reaction happen. So no ES complex is formed. Reversible competitive are the one which resembles like the substrate and bind at the normal active sites. They are like decoy. So they become the same shape of the substrate. They co-attach there, and action could be reversed, also could go back, and they act like a normal substrate. In the reversible non-competitive inhibitor, they usually binds not at the active site but somewhere else, which will inhibit the activation of substrate to that to the enzyme. So binding is weak, so this inhibition is reversible. Let's take one by one all these inhibitors. So first is irreversible inhibitors. So irreversible inhibitors, they bind very tightly to the enzyme, and the binding of the inhibitor to the R group, yeah, of amino acids in the active sites and this binding may block the active site. It will not let to attach any, does not make enzyme substrate complex, cannot form in this case. Alternatively, inhibitor may interfere with the catalytic group of the active site, eliminating catalysis. So irreversible inhibitors includes arsenic, snake worm, nerve gas. So they, once they are in our body, it will make our body just not to do anything. If we have even small nanogram amount of all these nerve gas, snake venom, arsenic, so our all the enzymatic reactions in our body, they will be instantly stopped, and nothing, we can't even help it out in order to save ourselves. Yeah. Now reversible competitive inhibitor. So in this reversible competitive enzyme inhibitors, they're also called as structural analogs, that molecules that resemble the structure and charge distributions of a natural substance for enzyme, and they resemble permits the inhibitors to occupy the enzyme site. So once inhibitor is at active sites, no reaction can occur and the enzyme activity is inhibited. So inhibition is a, so what we are seeing here basically, uh, they just act like a substrate, like decoy. So it doesn't let the substrate to attach to it, and then so that because of that, once the inhibitor is at the active site, no reaction can occur and enzyme activity is inhibited at the end. So in this inhibition, it is competitive because the inhibitor and the substrate, they are fighting for one site only, that is the active site. And the degree of this fight, degree of inhibition, will depend upon the concentrations of your enzyme and inhibitor. How much concentration of your inhibitor is present as compared to enzyme. So according to that, this competitive inhibition will work out. Then reversible non-competitive inhibitors, they are the one which enzyme inhibitors bind to R group of amino acids or to the metal line factors. And binding that is happening here is quite weak. Binding is not that strong. And enzyme activity is restored when the inhibitor dissociates from the enzyme inhibitor complex. And these inhibitors do not bind to the active site and do modify the shape of the active site once bound elsewhere in the structure. Next is your proteolytic enzymes. So let me just repeat what we have done since morning in these lectures so that uh, quickly revise everything from the start. We were discussing since morning about nomenclature, classification, enzyme substrate complex, cofactors and coenzymes, proenzymes. We have discussed 6 types of nomenclature, classifications of enzyme with the various examples of them. Then we discuss how we give the name to the enzymes with the A's at the end and according to the property that they're working. Then we discuss about the equilibrium constant, KEQ, with product raised to power B, reactant raised to power A. We discuss that the catalyzed reaction, um, it's lowered in the help of in the presence of enzyme. The activation energy is lowered, whereas in the uncatalyzed reaction it is quite high. Then we have discussed that in the presence of enzyme, in the rate of reaction versus substrate concentration, it's make a hyperbolic structure in the presence of enzyme, whereas in the absence, it's make a linear line. Then we discussed various states that are happening, that is enzyme plus substrate, enzyme substrate complex, transition state with enzyme product complex and then enzyme product. So we discussed a little bit about these details about active site, the various properties of the active site. We discussed 2 models, lock and key model and induced fit model. In the lock key model, basically it doesn't changes, the conformation changes doesn't happen. In the induced fit model, it happens. Then we discussed a little about more about the transition state, how it's make the reaction faster by seeing that there are two products. So glucose or fructose, they combine together and remove the water. So in this, enzyme is helping to make this in transition state, making this thing happen. And then we discussed about cofactors and coenzyme, that like copper here, if it is present, only then the reaction could be possible. And also we discussed that coenzyme could help to in order to transfer your one functional group from one substrate to another substrate. Various example of coenzymes, various vitamins we have discussed in our body. Then we have started the topic of allosteric enzyme. In allosteric enzyme, some they are mainly act as a negative inhibitor. Positive and negative allosterism is present. Um, so positive allosterism, it's mainly, yeah, effector. They get attached to the effector side, um, and then doesn't let your reaction happen by stopping your active site. That's how the negative allosterism will work. If it is getting attached and making reaction even faster, then it will be positive allosterism. So in this case, in this reaction, the third reaction of your glycolysis, if ATP is present, it will be negative effector. If it is AMP, then it will be positive effector. We discuss then further about feedback inhibition. In the feedback inhibition, we have discussed that if product F is produced during this reaction, it could attach to the enzyme into the negative effector site, act as a negative effector, and doesn't let reaction to happen again. So this will be a negative feedback inhibition. Same way, the positive feedback allosterism is possible when the hemoglobin is attached to the oxygen in order to attach more oxygen to himself to transfer oxygen to the lungs. Then we are discussing about proenzymes, which are in an active form and they get active in the presence of cells by proteolysis or hydrolysis of the enzyme. So some examples we have discussed in the digestive tract. Then we are discussing about protein modifications, and then the new topic we have started today is the inhibition of enzyme activity. So we have discussed mainly there are three types of it. One is irreversible, and under the irreversible there are two, competitive and non-competitive. The irreversible is the one which binds to the amino group to the substrate. So substrate is acting like the inhibitor is acting like a substrate, and it's get attached to it and doesn't let the reaction happen. So irreversible inhibitors includes arsenic, snake venom, nerve gas. So it doesn't let the reaction happen anymore. So no enzyme substrate complex is formed in that case. In this case, the inhibitor it somehow decoy the shape of the substrate, and it's attached to the active site, and also doesn't let the reaction happen. In non-competitive, it could be restored in the previous form, but it doesn't attach to the active site, it attaches to some another site, so it makes the reaction slow. Now proteolytic enzymes. So proteolytic enzymes, they are the one which cleave the peptide bonds in proteins. So in order to have the primary structure of protein, so these enzymes specificity depend upon the hydrophobic pocket. So cluster of hydrophobic amino acids that brought together by 3D folding of protein chain. So for example, chymotrypsin, which cleaves peptide bonds at the carboxylic end, so like methionine, tyrosine, tryptophan, phenylalanine. So we have alanine here, phenylalanine here, glycine here. So these peptide bond that is being made here, it could be break with the help of various enzymes like chymotrypsin. So some of the applications of these enzymes in medicines. So in diagnostic, first of all, to alter the with the disease. So in heart attack, like lactate dehydrogenase, creatinine phosphatase, SGOT. So we check all these features in order to find out the the reason for the heart attack, what are their values are. In pancreatitis, amylase and lipase activity we check. Then in analytical reagents, enzyme used to measure another substance. Then urea is converted to ammonia via urease, and blood urea nitrogen is measured. Then replacement therapy. So administrator genetically engineered the glucosaminidase for Gaucher disease. So these are some references. Thank you very much. You can contact me on this email ID if you want. I will share this notes with you now. Okay, this is the actions, right? So this is the second part that I'm sharing with you. So students, just give me 5 minutes break. So it's 18:15 or 13. We meet around 18:20 now. Okay, this is done. So the next thing, so we have discussed in quite specific manner the general properties of enzyme. We have discussed the inhibition of enzyme that was not required in your course, but I did. We have discussed quite in details about mechanism of enzyme actions with proenzymes, their activation, allosteric enzymes. So most of your topics are being covered in between. We have discussed lock and key model, induced fit model. Yeah. Um, so most of your topics are being covered here. We have discussed this ES complex, binding site, active sites. So all these topics are being covered today. General properties are discussed. So majority of your 60 to 70%, 50% course is done. Now I wanna take you to the next level. It's not written in your course title, but I will like to take you to the enzyme kinetics, which is very essential in order to, if you are having enzyme engineering course, enzyme kinetics will be very important for all of us. So did I share the history also with you or not? So I shared with you the book so far. Objectives were shared, book was shared, not the history actually. It's okay, it's from my Rama University, you can have it. There are some handwritten notes from my students from the unit 1. You can also go through them. They are pretty good. So whatever I have described you, they are written in very nice forms. So you can also have a look over it. So around it's 18, 20. We meet at 18, 8:25 now. Yeah, just give me 5 minutes break. Make myself ready for the next session, and then we start again. Cool. So, welcome back. Just a second. So, let's start with the and students. One thing I want to tell you that tomorrow we will not have the class 7 a.m. in the morning. I will have another classes at that time. So we will meet today from 3 p.m. to 5 p.m. Okay. So now I want to take you the next journey. So we are done with the major topics for today. Nomenclature, classification, mechanism. Now it's the enzyme kinetics. So in the enzyme kinetics, before we start upon enzyme kinetics, just rebrush our memory. So this we have seen in the first lecture, that enzyme plus substrate make an enzyme ES complex, and ES further have a transition state, makes EP, and then we have our product. Yeah. So let's check how these reactions happen from the perspective of kinetics. So here we can see that um, substrate concentration and reaction rate. So on the left hand side, we can see the maximum velocity, that is V initial reaction rate with a mole per liter per second. So, and on the bottom is the concentration substrate, mole per liter. So what is happening is, initially the enzyme will have very less substrate, so few will attach. In the middle range, we will have good number of substrate, so all will be attached to the enzyme. By the end, there will be lot of substrate, so enzyme is quite saturated and it cannot take any more substrates at the end. So it's get to the maximum velocity. So here enzyme activity increases and all the enzymes are combined with the substrate, and at the end we have the maximum activity. So that's the basic scenario behind your enzyme and a substrate working together during the reaction rate. So the rate of reaction increases as substrate concentration increases at constant enzyme concentrations, and the maximum activity occurs when the enzyme is saturated. So when all the enzymes are binding substrate. So here we can see, as we have discussed also, what is the active site. So active sites is the something on which substrate is attached towards enzyme, and there could be some conformational changes or some things could be happen in order to make the binding stronger. So the active site consists of residues that form temporary bonds with the substrates and residues that catalyze the reactions of that substrate. Although the active site is small relative to the whole volume of enzyme, and it is the most important part of the enzyme as it directly catalyzes the chemical reactions. So it usually consists of 3 to 4 amino acids, while the other amino acids within the proteins are required to maintain the protein tertiary structure of the enzyme. So you have your active site here, and the enzyme being substrate being attached to it, and the products are being formed later. So what are the units of enzyme activities according to the international unit-based system? So enzyme units are never expressed in the terms of milligram or microgram. They're always expressed as activity. And to maintain the uniformity world over the enzyme activity, so we have brought a new unit system according to IUB system. So this is the classical unit that we use for enzyme activity. So there are three activities we have to consider, that is enzyme activity, specific activity, and molar activity. The enzyme activity could be calculated as micromolar, yeah, micromolar substrate transformed. So mainly per minute. So the rate of reaction would be governed here, like how much in 1 minute, how many of substrate are being transformed into product. That's how the unit of our enzyme activity is calculated. So we call it as a unit. So unit is called as micromolar substrate transformed per minute. And the specific activity, it's calculated as micromolar substrate per minute milligram of enzyme. So unit milligram, unit per milligram enzyme, and molecular would be micromolar substrate, that is a unit per minute of your micromolar enzyme. So unit per micromolar enzyme. So basically, activity unit is what is internationally it is called as actually Catal. So it's the catalytic of enzyme is depending upon the specific conditions such as pH, ionic strength, buffer type, presence of cofactors, and suitable temperature. An international system of units designated molar per second and is recommended as Catal. उसको हम Catal कहते हैं. We call it as a Catal. and microcatal is equal to 10 raised to the power minus 6 catal, that is your micromolar per second. Catal could be calculated as a micromolar per second. So concentration, if you want to find out the concentration of enzyme activity, so we can calculate as a micromolar catal per liter if you want to calculate the concentration. So following units are derived. So as we have discussed, specific catalytic activity and molar catalytic activity. Yeah, so these are the three activity centers that we are working on: specific catalytic activity and molar catalytic activity. So specific catalytic activity is the activity of enzyme preparation in relation to the protein concentration, and molar catalytic activity, this can be determined with the pure enzymes with the known molecular weight available. So it is expressed as catal per molecule per mole of enzymes. That is catal per mole of enzymes. So remember that, and the new term that I want to show you is the turnover number that was residing in your syllabus. That what is turnover number? So turnover number is nothing. It's an enzyme that has only one active site per molecule, and molar catalytic activity equals to the turnover number, and it is defined as the number of substrate molecules converted per unit by each active site of the enzyme molecule. So I will repeat, the number of substrate molecules converted per unit time by each active site of the enzyme molecule. So how much your product is being produced per minute, that is known as your turnover number. So let's talk about each enzyme activity, specific activity, and turnover number, these three definitions. So enzymatic activity, reaction rate is expressed as a change in concentrations per unit time, that is mole per liter per second, and for enzyme catalysis reaction, substrate is turnover per unit time is commonly used. So catal unit is a mole of substrate per substrate, which is micro catal, and the international unit is micromolar of substrates per minute. And 1 catal is equal to 6 into 10 raised to the power 7 unit. But the specific activity is a unit of enzyme activity per milligram of protein, and 1 unit is equal to amount of enzyme that will convert 1 micromolar of substrate to product in 1 minute at a given pH and temperature. And we calculate them at the purity of the enzyme to calculate the specific activity. And then for example, you have to identify there is 5 milligram of proteins in an extract which is catalyzed to make 100 micromoles of substrate to product in 10 minutes. So what is the specific activity of that enzyme? The answer would be 2 units per milligram of protein. Then turnover number of an enzyme, which is also termed as K cart. So this is defined as the maximum number of chemical conversions of substrate molecules per second that a single catalytic site will execute for given enzyme concentration. So K catal will be equals to V max divided by ET. So you can calculate this turnover number by this calculations. For example, carbonic anhydrase, the turnover number is around 600,000 per second, which means that carbonic anhydrase will produce around 600,000 molecules of products per seconds. So here some example calculations of turnover number. That is, an enzyme is 1.84 gram and the molecular weight is 36,800, and we have to, in the presence of a substrate, excess substrate that catalyze at the rate of 4.2 molar substrate per minute. So we have to calculate the turnover number, that is mole substrate per minute per molar enzyme. So in order to calculate that, first we have to identify mole of enzyme that is your enzyme activity. So for that you have to divide the gram that is given, 18.4 gram, divided by the molecular weight. So in this way you have the molar enzyme activity 5 into 10 raised to power minus 5. Then to calculate the turnover number, as we have discussed previously, you can divide Vmax by ET. The Vmax we are already given 4.2, and we have calculated the ET. You divide with that, and then you get the value of 84,000 per minute to find out the. So 84,000 per minute is the turnover number, that is this enzyme will convert the substrate into the product per minute 84,000 times. And same some examples for like catalase, carbonic anhydrase, acetylcholinesterase, chymotrypsin, lysozymes. So all they have the specific turnover numbers with the various Michelson-Menten equations. So catalase has conversion of H2O2 to H2 and O2. Then carbonic anhydrase, hydration of carbon dioxide. So various example with the specific turnover numbers. So So kcat is the first order rate constant describing your enzyme substrate into enzyme plus product, which is also known as a turnover number because it describes the number of reactions that are happening in a molecule of enzyme and catalyze the reactions per second under optimal conditions. So most enzymes have kcat values between 10 raised to power 2 to 10 raised to power 3 per seconds. So here we can calculate these values. So we know this E plus S give rise to ES and ES give rise to E plus P. So the K1 would be the first equilibrium constant, the Kcat will be the another one, and K minus the reversible one. So according to the Michelson-Menten equation, we will make this hyperbolic reaction. This will be the region B, region A. So we will discuss about this in detail. So here this one is K1 is there, K2 is there, K3 is this one, and in order to find out the Michelson-Menten equation, so there was a hyperbolic saturation kinetics have been created. So this is the relation between the substrate concentration and velocity of a reaction. So what we do is we have half we create this hyperbolic structure. So on the x-axis is the substrate concentration, on the y-axis the reaction rate. So as this reaction is going up, so at some point it will stop, and oops, it will form the maximum reaction to be Vmax, but thing is it will never reach to the maximum reaction. But the time we get this half Vmax here, it will be corresponding to the constant Michelson-Menten constant Km here from here. So in this way we calculate this value. So Michaelis-Menten equation, how do we represent this? V naught is equal to Vmax multiplied by substrate divided by Km plus S. So whereas V naught is the velocity of the reaction, Vmax is the maximum velocity of reaction. So V naught could be any value here, like initial reaction, and Vmax is the maximum where it reaches, this one. And then so S is your substrate concentration and Km is your Michaelis constant. So this is quite important, very important perspective for Michelson-Menten equation. So Km actually it's a ratio constant, ratio ratio of rate constant, that is K2 plus K3 by K1, and it is equal to the substrate when initial rate of V is equal to half Vmax. And it's a property of ES complex, does not depend upon the concentration of your E or S. And whereas the Vmax is the maximum velocity at fixed E and directly proportional to the enzyme. Okay. So these were the Michaelis and Menton. They discovered these equations together. So rate reaction being on the y-axis and substrate concentration on the x-axis, and yeah, Km on the bottom, Vmax like this. So Vmax is approached but never achieved. So that's one thing that happened with this kinetics. It never achieves the Vmax. So I think I will stop here. We will stop from this perspective, and we continue in the next lectures, because I can take some queries if you have now, because this will be too much in continuing, because we are starting from 7 a.m. in the morning and we did a couple of topics one on one. So we should save something for the later also and take some rest at the moment, and we continue with the Lineweaver work plot, and we will discuss about some uncompetitive inhibitor, competitive inhibitors from the kinetics perspective. Yeah, and then I will take you to the isolation of enzyme and purification of enzymes, how we do it, what are the various techniques are available. So we will discuss about them in detail also. Yeah, so thank you very much till here, and we see us at 3 p.m. sharp. I will share the link with you, because I heard that there's a 1 to 3 p.m. is your vaccine class. So based on that, I can't take your further classes. So yeah, um, we see us then at 3 p.m. Thank you very much. Take care. Bye bye.